Good afternoon, everyone. I was going to start to present myself, but it's already did. My name is Milan, and I'm French, but I'm doing my PhD here in Canada under the supervision of Professor Arnaud Droit. So I'm going to present the results of a paper that was published this June in Computational and Structural Biotechnology Journal called Target Repositioning Using Multilayer Networks and Machine Learning in the Case of Prostate Cancer. So first, I'm going to define a few terms. What is target repositioning? So many of you might know what drug repositioning is, which is when you know that a drug can cure a certain disease based on its properties and the properties of this disease. You can maybe infer that this same drug will have an effect on another condition. And so this is drug repositioning. Target repositioning is the same thing, but with protein targets. So if you have a protein that you know is highly involved in a disease or targeting it will have therapeutic effects against this disease, we can maybe infer that the same targets targeted by the same drugs or other drugs could have a therapeutic benefits against another disease. And so the main advantage of target repositioning is that by discovering new protein to targets, we might attack different pathways and therefore combat resistances within the disease. Then why prostate cancer? So prostate cancer is the second most common cancer in Canada. Its survival rate for non-localized prostate cancer is less than 32% 5 years after diagnosis. And the main reason for this is the development of resistances. So I put to the right the main biological pathway, obviously summarized in prostate cancer. So the LH and FSH hormones are released by the hypothalamus and in the testis, which leads to the conversion of cholesterol through a series of cascades into testosterone, which is the main androgen hormone, which activates the androgen receptor, which leads to the stimulation of a series of genes that are important for progression of normal tissues, but also of prostate cancer tissue. And so we have a series of treatments that can block this pathway on several points. And they work for a couple of time at least, but eventually the tumor always creates resistances, either by amplification of signals using different reactions or mutating key proteins. And so we need to find new proteins to target and new treatments. And there are lots of work doing right now doing research. And so the last key treatment is PARP inhibitors, which show promises, but obviously we're still looking for cures. And so we need to find new proteins that we can target and not new drugs that target the same proteins. And thus the need for target repositioning. And so this was the introduction, and now it's the goal to find these new proteins. And so I'm doing classical machine learning, supervised machine learning. So I'm trying to fill this database. So I have my protein targets that I obtained from the Pharos databases, which are all the proteins that we know can be targeted by drug-like molecules. And I build their class, which I obtained from Blood Bank, which basically means if a protein can be targeted by a drug-like molecule that is either currently investigated against prostate cancer or already marketed against prostate cancer. So this protein will be labeled as positive and the other one as negative. And now the goal is to find features for this data set. And so I'm going to use a network. So we built a large multi-layer network by integrating several biological databases that encompass protein-protein interaction networks, transcription factor to gene regulations, microRNA to gene regulations, molecular pathways, GO-terms, and metabolites interactions. And so all of this data forms a 5-layer network, which is quite large and contains more than 60,000 nodes and more than 2 million interactions. And all of these interactions are experimentally validated. So now the question is how to analyze such a big network. So what people usually do in target repositioning when they want to find new targets for a specific disease is, for example, if we try to extract features for the protein that is marked by a star, is to assess if it is close or not close to the cancer signature. So for example, if we know that a couple of genes are mutated in this disease or differentially expressed, we can maybe look in the network how close a protein is to that signature. And the closest it is, the better it has of producing a therapeutic effect when targeted. And so this is usually what is done using different proximity measures, obviously, to find new proteins. But there are also other ways, and maybe we could talk about biases later, but I promise I took that into account, even if I'm not presenting it right now, based on, for example, similarity and topological metrics. So I was looking like, if a protein is similar to another protein, like the receptor, the androgen receptor, maybe it's features that is worthwhile to include in the model. And if it is far away from the network, it's not going to be similar. And I also extracted topological metrics such as degree and betweenness and centrality. I also looked at modules, that is, communities of nodes densely connected within the network, and maybe being part of such a module or being close or far away from this module can be a useful information to predict therapeutic targets in the case of prostate cancer. And finally, propagation-based methods, which basically calculate distance between my star protein and other nodes in the network in either a downstream way, so from my protein to another node, or in an upstream way, from the node to the protein, which can be different because there is directed interactions in the network. And so after extracting all of these features, I end up with more than 460,000 features. So it's quite a lot of features. Here, I'm not going to go into too much details, but I have actually 11 data sets. Each data set represents a type of features that I'm going to analyze separately so that I can calculate how well the features extracted are in predicting what we know already about therapeutical targets. And so for each of these 11 data sets, I run several machine learning models, random forests, support vector machines, can-nearest neighbors, naive Bayes, and neural network. I fine-tune them through several cross-validations in order to find the best machine learning algorithm, but also how these different features are useful in predicting known therapeutic targets. And so here, I'm not going to go into too much details because there was a lot of information that you can find out by, and that is in a paper, such as, well, if you want to know if signature-based methods are predictive, you can use mutated genes, like maybe being close to mutated genes is good, but maybe it's differentially expressed genes or structural variant genes. So we could find out some results like this, or what layer is most important in the graph, or is being directional important or not? Is it better to have non-directional interactions in the network? But here I'm just going to focus on the features that I presented in the introduction, which are these four, with the MCC as a natural symmetric on an independent test set. And so as you can see, module-based and signature-based methods were did not have good performances. And it's maybe difficult to understand why, because these modules were like, the algorithms that find the modules were not mistaken. There were modules, maybe they were biologically relevant to the prostate in general, but being close or being part of those modules did not lead to good classification accuracy. So even if they were biologically relevant, they did not help in differentiating therapeutic targets to non-therapeutic targets. And most surprisingly, these signature-based features also did not lead to good performances, which is surprising because the bulk of the papers published in target repositioning use this method. And so, again, it's not clear why. Maybe it's not clear why, but focusing only on proteins that are close to the signatures did not lead to good classification results. So the best two type of features here were propagation and similarity-based methods, which were then used for the final prediction of targets. So I used the best model, which was the can-nearest neighbors, and I trained it on every target that I had. And so the targets are in an unbalanced fashion, so I have more negative than positive, and the KNN would try to separate the two. And so sometimes he's going to make a mistake. He's going to think that negatively labeled target is actually a positive. It's going to be a false positive. But if I'm doing this repeatedly across several cross-validations and with the two types of features, I realized that some false positives appeared constantly. The KNN algorithm could not predict it as a negative for some reasons. And so the hypothesis is that those false positives were not supposed to be labeled as negative and were my real prediction. And because obviously, if I labeled at first my positive targets as positive, the other one are in fact truly unlabeled. And so this was the way of finding back across the unlabeled targets, the ones that had the most chances of becoming true positives. And so doing that, I obtained 10 new targets to be validated. Sadly, validating targets in vivo is difficult, and we could not do it in vivo. We did not try. We don't have access to the in vivo facilities, so I had to validate them using the literature. And so among those 10 predicted therapeutic targets, it appears that the first 5 were actually already investigated. So that means they are quite researched in prostate cancer, and they have known drug-like molecules that can interact with them and showed therapeutic benefits. So this one, the algorithm find a known truth about targets. That's potentially, if I could rerun it again, would be initially would be labeled back as positive. And the other 5, well, the other 4, the next 4 are what I call validated. That means they don't have a known drug-like molecules that showed therapeutic benefits, but they were highly studied in the case of prostate cancer, all involving in different pathways that were also studied in prostate cancer. And so they show therapeutic potential and could be potentially more investigated. And the last one, which is NPBWR1, is relatively unknown. So it has very few research on it. So it was difficult to understand why the model predicted that protein and not someone else, some other protein. And so by looking at the features that were expected, which were based on the topology of the network, and so we could infer that this protein was highly similar to 2 other proteins, because that similarity was chosen by the model. These 2 other proteins were GNA1 and 2. And these 2 proteins are opioid receptors, and they are both also involved in prostate cancer, including its metastatic properties, its invasion and migration properties as well. So NPBR1, which is almost involved in the same pathways within the network than those 2, we infer that that's the reason why it was predicted. And so to conclude this presentation, we have developed a method that automatically extracts many network-based features from a complex biological network that can be used for repositioning. Obviously, it has some limits, including that the predictions are difficult to validate biologically because, well, it's difficult to test many different drugs and show that none of these drugs works on these targets. And also that the results that I show that, including the variation into predictiveness from different features, could very well and probably are specific to the disease and the network and the data. And so to compensate that limit, we created a R package called Disconnect that has the same function that extracts all the features with some feature selection methods, so that any of you could reproduce that study on your network and on your repositioning case, including drug repositioning if you want. And so the key point to take home is that target repositioning is an underexplored strategy, at least compared to drug repositioning, and that showed that it could find back known targets, and not to rely on only one method, but try several. Because if I were only to focus on signature-based strategies, like the most paper did, I would not have achieved the best performances. Thank you for listening.
We have time for some questions. If people have questions, feel free to get up to the microphone.
Thank you very much for the talk. I'm sorry if I missed it, and you already have mentioned that, but I'm curious, how did you connect genes, proteins, and metabolites. I don't remember exactly. Like how did you connect the entities or nodes across layers?
Yeah. So because it was like directional, proteins to genes were based on transcription factor regulating genes. So it was a downstream arrow from the protein to the gene, and the other arrows were based on what kind of like the gene once translating did create protein products. And so this was another arrow from just what kind of protein did the gene gives when translated. And from the metabolites, it was based on like a reactome or keg, which has already pathways where you can extract those pathways, which contain genes, proteins, and metabolites. And I just added them to the network.
Okay, thank you very much. Thank you.
Hi, thank you. I'm not sure if you included genetic knockout, knockdown data in your training data set, but if not, then would this be something you could potentially consider for validation? So look at siRNA or CRISPR data sets in prostate cancer models.
Yeah, I could probably do that. But not personally, because we only focused on bioinformatics. But looking at the literature, most of the targets I found were validated. I did not show all the results except the last one, which was only validated because like its methylation pattern was connected to bad diagnosis. But except that, I found nothing. So the other one were already validated. But sure, I could interpret this for like more powerful validation.
Yeah, thank you for the presentation. So I wanted to ask just two questions. In terms of the network connection layers, did you kind of concatenate the layers, or you just use independent networks for each of the layers? And also, will it also be kind of good to include single cell data and try to see how you predict, and also the database you use for target prediction? I don't know what database you use. Just thank you.
Well, yes, they are all connected, so you can go from one to the other following paths. For the single cell, do you mean like inferring a new network from the single cell data and integrating it?
Or maybe just adding it as a feature.
Oh, yes. So, yeah, I agree. It could boost the performances for sure. But I was only also looking at if only the biological networks, so just experimentally validated interactions, could suffice in predicting the therapeutic properties of targets, which is mitigated recently.
So the internal implementation, is it kind of concatenating, or I mean, I'll probably just check it later in your GitHub, and also the therapeutic database, which do you use
to
validate the targets?
Oh, I use the scientific literature that was published. So I was looking at the protein and the papers that were published. And if they said it was investigated, if they said that they proposed a new drug for it, I validated it like that.
Okay, thank you.
Thank you.
Yeah.